Hello to you all. My name is Noma Assad, and I'm a PhD student in my second year. And I'm going to present to you my ongoing project about dipeptidyl 3 or DPP3. It is preclinical data. So to start with, what do we know about DPP3? Actually, it is a metallopeptidase, which cleaves dipeptides from the amino terminus from several substrates, such as angiotensin. It is ubiquitous, expressed in all of the tissues and organs. It is intracellular, but it has also been detected in the circulation of healthy individuals and with increased concentrations in critically ill patients. So this increased circulation of circulating DPP3 or cDPP3 were associated with decreased survival in ICU patients with cardiogenic shock, septic shock, or hemorrhagic shock, which shows DPP3 as a biomarker with prognostic interest. So in our project, what we want is to go a step back and try to understand how circulating DPP3 is increased in the plasma of these patients. So we posed several hypotheses. The first one is that the tissues are increasing expression of DPP3 in these pathological situations for some reasons. The second hypothesis are that they are increasing the release of DPP3. And the third one is that there's an accumulation of DPP3 due to decreased clearance. I will concentrate more on the first two hypotheses. Now, in order to study the expression of DPP3, we use two different mouse models of acute cardiac dysfunction, pharmacologically induced by either injection of isoproteinol in the first model on the left, ISO model, or by injecting of doxorubicin, the DOX model. At the end of each model, we collect plasma samples and we harvest the organs. Now, what we have seen that there was a significant increase in the activity of cDPP3 in mice with acute cardiac dysfunction compared to control mice in both models, which go in parallel with the clinical data and with previous preclinical data. Now, moving to the expression of DPP3, we wanted to take an exhaustive look of the expression of DPP3, not only in one organ, all of the organs, on mRNA level and on the protein level. So here in this slide, I show you only the organs where there was a significant increase in the expression of DPP3. So in the ISO model, on the mRNA level, we have increase in the heart. On the protein level, we have increase only on the bone marrow cells. However, in the DOX model, we didn't have increase in mRNA level in none of the organs, but we have seen an increase on the protein level in the spleen. So what this result shows us is that, first of all, we see that there is an overexpression in this pathological situation in both models, that the expression varies, so we don't have the same organs that the same increase in both models. So it varies the expression based on the mechanistic or based on the model used to induce the cardiac dysfunction. And we've seen also that there was no correlation between mRNA and the protein expression. However, on the other hand, we have seen some organs that there was a decrease in the expression of DPP3, which leads us to the second hypothesis, which is the release from tissues, meaning that this tissular decrease is because there had been before a release into the circulation. And when we think about release from cells, the first thing comes to mind is the cell death. So in order to study the cell death, we worked in vitro, starting with cancer cells, which is a little far from our pathology, but it was a proof of concept because they highly express DPP3. And then we passed to another cell line, which is human cardiomyocyte, AC16. What we do is that we subject the cells to different stress conditions, and then we evaluate the amount of DPP3 released in the supernatant of cell culture around these cells. So we can see here the mortality of cancer cells. Starvation means that we decrease the nutrient in the medium. DMEM, there's no nutrient, so we starve the cells. And then the doxorubicin in increased doses. We can see here that there's an increase in mortality, logically. And in parallel, we have an increase in the release of DPP3 in the supernatant, which leads us to this very obvious, positive, significant correlation between cell mortality and the levels of DPP3 released in the supernatant. However, so meaning that cell death participate in the release of DPP3. However, when we go to the cardiomyocyte, we don't see this clear. It's a weak correlation, barely there's no correlation. And this reflects that, first of all, it depends on the cell type, the way that this protein is released. Second of all, it suggests new release mechanisms next to cell death. Now, speaking of this new or different cell mechanisms, we have the shedding. And before I explain the shedding mechanism, just a few idea about the localization of DPP3. So mainly DPP3 is a cytosolic. If we see here in the table in data about proteins that has 0 transmembrane helix, it has no signal peptide, which is a hydrophobic sequence that allow the protein to be in the membrane. So what we know is that it is cytosolic. However, there is studies that shown otherwise. For example, here is a study about a mouse model of Alzheimer's disease where they analyzed lipid rafts that are in the membrane of brain cells, brain tissue. And when they analyzed this lipid raft, they saw DPP3 in the extract of these membrane structures. And when we did immunofluorescence, we see DPP3 a little bit on the membrane. If you see, there's a cardiac tissue, DPP3 in green. So we wanted to investigate the shedding mechanism. So the idea is we have enzymes that call SHEDAs, a group of enzymes that cut the transmembrane proteins and release the extracellular portion of the enzyme into the circulation as a soluble form. So we hypothesize that DPP3, for some reason, goes to stress, it has modification in the structure, and it's taken into the membrane and then shedded by this enzyme. So what we did, that we used the TAPI2, which is a broad spectrum inhibitor of these enzymes, and then we wanted to verify what happens on the circulation of DPP3 in the supernatant. And logically, if the hypothesis is correct, they should decrease when we use the inhibitor. But actually, this is not what we saw. There was no significant difference between when we stress the cells alone with doxorubicin or we stress them and before we put this inhibitor. There's no significant. So at least in this context, in this cell line, we don't have the shedding participating in the release of this protein. So to conclude, we have seen that the increasing expression, the cell death, participate in this increase in circulating DPP3, and that the expression varies based on the model. For our next step, it will be to investigate in the extracellular vesicles as a mechanism suggested for the release of DPP3, and to study also the hypothesis of decreased clearance. Thank you very much.
Time for questions.
Thank you very much. That was an excellent talk. Just wondering, have you actually do a little bit of phenotyping of your cell types, looking at the tissue expression of the DPP3, and then actually pinpoint which cell type you should be looking at in the first place, and then later on the cell culture experiment could be more focused on the cell type that could be affected in the first place?
You mean if we studied before if these cells express DPP3?
Exactly.
Yeah, everything we worked on, we study before the expression in the pellet of cells before we start analyzing the supernatant and the release. We have it in cancer cells, different types. We have it in cardiomyocyte human, and we have it in rat cardiomyocyte. At least these are the cells that we worked on the lab, and the next step will be endothelial cells that we're going to work on.
I guess what I'm interested in is when you look at the heart tissue itself, have you phenotyped every single cell type? For example, cardiomyocytes, endothelial cells, smooth muscle cells, pericytes, for example. Look them all and see which one is actually affected, and then you can then focus on that cell type.
Yeah, it is a good idea. We did a study on the tissue as a whole, and we did on just cardiomyocyte for now. But yeah, for the future, we should do all of the cells in the cardiac tissue.
Perfect. Thank you.
Thank you, Noma.